Okay, so we are going to start our genetics portion from this chapter onward, and we will slowly build our base, and the base will start from chapter number 7, that is about DNA, RNA and central dogma. This chapter is going to be really important because it will create your basics, and then on those basics we will start building up your whole concepts. And this chapter is actually a bridge between your previous chapters and the chapters that you will study in your future, like chapter number 2 about cellular organelles, and chapter number 8 about DNA replication, transcription, translation, and even about genetics and inheritance. Those chapters will be really integrated with this chapter. Even when you go towards the cell cycle chapter, this chapter will be your like the base, and these all chapters are connected with each other, so you don't have to leave any of these topics, because if you leave this chapter and these topics, and your base will be like really weak, and without your base, you can't go into complex topics and difficult concepts that are hard to grasp if you don't understand the base. So in this chapter you will discuss about DNA, about its structures, about nucleotides, what is that comparison between DNA and RNA, about base pairings, what is central dogma, types of RNAs, chromatin and chromosome structures, prokaryotic and eukaryotic DNA. So these are the topics that we will be discussing in the first introductory chapter, and these are the high yield concepts that you have to know. Take a screenshot, and then we will move on to our first topic. So let's start with our first topic, that is about nucleotide structure. So nucleotide is a monomer of nucleic acid. So we have already discussed about nucleotide structure in our previous lectures in biological molecules, or you can say chemistry of life. So in that chapter we discussed about nucleotide structure in detail. We will be revising that, and you already know that the nucleotide is made up of 3 components: a sugar, a phosphate group and nitrogenous base. So sugar, as you can see here, is the central portion, and sugar is ribose sugar, right? And ribose sugar is present in RNA, but if there is one less oxygen, then it is called as deoxyribose sugar, and the deoxyribose sugar is present in DNA. So there are 2 types of sugars, one is ribose and the other is deoxyribose. Ribose sugar follow the conventional basic formula for carbohydrates, but deoxyribose is an exception to the basic formula of carbohydrates. You have to remember that, because deoxyribose has one less oxygen than normal basic formula for carbohydrates. You know that what is the basic formula, that is CnH2nO, but deoxyribose has one less oxygen. That's why it does not follow the basic conventional formula for carbohydrates. The other group is, the first group is phosphate group. So phosphate group, as you know, has a negative charge, and because of phosphate group there is negative charge on our DNA and RNA, and phosphate group is attached with the carbon number 5, as when you start counting 1, 2, 3 and 4 and 5. So phosphate group is attached with the carbon number 5, and nitrogenous base is attached with the first carbon. And as you know that there are different types of nitrogenous bases, we will discuss about them, and they are purines and pyrimidines. So the first is purine, the other is pyrimidine. If you can see here that purine actually has a smaller name, right? Purine, but it has bigger structures, while pyrimidine has bigger name but it has smaller structures. So first of all we discuss about purines. So the first purine is adenine, and then second is guanine. So purine and guanine has approximately same structures. You don't have to memorize every single atom and every single structural formula. You just have to know the differences between them. For example, if you have memorized the structure of adenine, you just have to replace amino group with oxygen, and you have to add another extra amino group here and add extra hydrogen here to balance out the double bond that we had here. So you only have to memorize the adenine formula, and then you can just switch the amino group with oxygen and add another amino group in the carbon number 3. So that was all about purine structures. So you have to memorize only one structure. Similarly, you only have to memorize only one single pyrimidine structure. You can memorize thiamine, cytosine, or you can memorize uracil. It's up to you. So what is the differences between them? As you can see here, thiamine actually contains oxygen, cytosine contains amino group. So you can say that thiamine and uracil have same group, that is oxygen. So they both of them are same to each other, while you can see that thiamine and uracil have all of them have amino group here, that is on the base, amino, NH, NH and NH. But the difference between uracil and thiamine is that thiamine does not contain the same structure as uracil. They have basic structural differences. You know that there is a bond between adenine and thymine in DNA, but there is no bond between adenine and uracil in DNA, but adenine and uracil is present in RNA. So what is the difference here? Thymine actually contains CH3 methyl group, while uracil does not contain that CH3 group. So technically all of these pyrimidines are approximately the same, but they have just simple minor differences that we just pointed out. So if you just memorize one structure, then other structures would follow the same. So adenine and thymine are present in DNA molecule. If we make DNA here, then it will have a structure like this at GC. So they will have double bond, they have triple bond. We will discuss about it in our next topic, but for now you have to know that DNA actually contains adenine and thymine, while if we talk about RNA, then RNA contains adenine and uracil, while guanine and cytosine are the same. So that is the difference between nitrogenous bases of DNA and RNA. So there are two purines, that are adenine and guanine. As you can see, purine has smaller name but the structures are big, while pyrimidine has a bigger name but the structures are small and single ringed. So you can memorize them as the memory trick is pure as gold. So purines actually contains adenine and guanine. So purines are pure as gold. Purines equals to as gold, that is adenine and guanine. That is the memory trick. And pyrimidines, you can use the name that is cut, you know, CUT, cut. Cut for C for cytosine, U for uracil and T for thiamine. So cut is pyrimidines. So that is the memory trick that you can use. So that was the basic differences between purines, pyrimidines, and they were really important. So now you know like what is the basic structure. Now we will move on to our next concept, that is Chargaff's rule and base pairing. So Chargaff's rule has been really important before MUR era, like before 2023. This rule was really important. Like every year there was a question from this specific rule, but for 2 years, from 2024 2025, there has been no question from this topic. But I think this topic is still really important, because in future, like in next years, there might be some questions from this topic too. So you have to understand this concept by heart, because it's not that even hard, right? What does even it says? It says that there is a equal amount of nitrogenous bases in DNA. The equal amount is like, if you have adenine, then if you have 20 adenines, sorry about that, if you have 20 adenines, then you will also have 20 thymine. If you have 30 guanines, then you will have 30 cytosines. That is what Chargaff's rule is saying. Like it is not difficult, right? You just memorized it. So percentage of adenine is always equal to percentage of thymine, while percentage of guanine is always equal to percentage of cytosine. And if, for example, if we say if you have 25% adenine, then you will have 25% thiamine. If you have 25% guanine, then you will have 25% cytosine. And the sum of all of these, like 25 plus 25 plus 25 plus 25, you will have 100% total. So this is the rule here that is in front of you. Like if you have 25% of both of adenine and thymine, like let me give you an example. Like for example, if you say the sum of adenine and thymine is 25%, for example, if adenine is 12.5% and thymine is 12.5%, then the total would be, you can say 25%, right? And 25%, then you will need guanine and cytosine should be 75%, because the total should always be 100%. So if you know the percentage of one, then you can just simply predict the percentage of other molecules. For example, the example that is in front of you is, if you are told that there is 30% of base in DNA or adenine, right? So if you know that the adenine is 30%, so if adenine is 30%, adenine is always equal to guanine. So if adenine is 30%, then guanine would be 30%. Then both of them would cover out 60%, right? Then what is the remaining amount? That is 40%. So 40%, the remaining would be what it would be? It would be, sorry, it is adenine and thymine. So remaining would be guanine and cytosine. So guanine plus cytosine, remaining would be 40%. So how both of them contain 40%? If 20% is guanine and 20% is cytosine, then both of them should contain 40%. So it is, these kind of MCQs might be asked in IMAT. So as you can see here, they were really easy and you can like memorize them if you want to, but I don't think there is it is really necessary for you to memorize. If you have understood the concept, just repeat it again and again, do some practice, and you are good to go with this concept. So the next thing is about DNA double helix structure. So this structure was given by Watson and Crick, and the building of X-ray crystallography was given by the data from Rosalind Franklin. So what are the key features? The key features are in front of you. The saying is that sugar and the phosphate, as you can see here, are creating the backbone of the DNA here. So the backbone of the DNA is being made by phosphate and DNA. As you can see, this is the backbone of the DNA, while the nitrogenous bases are in the centers and they are forming bonds. So how many bonds are being formed? So adenine and thymine forms 2 bonds, while guanine and cytosine forms 3 bonds. We have already discussed it. We will keep repeating it again and again, so you don't make this mistake in your IMAT exam. And the other thing is, there are actually 2 types of grooves. There is a major groove, as you can see here. This is a groove and this is a groove, and again this is a groove and this is a groove. So as you can see here, this is a major groove and this is a minor groove. Then again this is a major groove and then again it is a minor groove. So the distance between a major and a minor groove is 3.4 nanometer. You have to know that that is the single turn. The single turn of DNA that is between major and minor groove is 3.4 nanometer. And if you talk about the width, or you can say that is 2 nanometer. And the distance between 2 base pairs like adenine and thymine and guanine and cytosine, the distance between both of these base pairings is 0.34 nanometer. So you don't have to confuse between 3.4 and 0.34 nanometer. 3.4 nanometer is the distance of whole turn. Whole turn means 1 major groove and 1 minor groove. That is the distance. So you have to know that 0.34 nanometer is the distance between 2 base pairs, 2 nitrogenous base pairs. So that is the simple MCQs that you have to memorize them. They are really important, and like don't mess them up. Okay, so next is the properties for DNA. As you can see here, one strand moves from 5' end to 3' end, and the other end ends from 3' to 5'. These are the properties that we have already discussed, so you can just read them on your own. So let's discuss about the comparison between DNA and RNA. That is really important. So first of all, you have to know that the DNA contains deoxyribose sugar and RNA contains ribose sugar. We have already discussed it. And the next is, what are the base pairs? You know that, as you can see in the diagram here, RNA contains adenine and uracil. We have already discussed it, we are discussing it again. While DNA contains adenine and thymine, that is the other basic structure. The other thing is, as you can see here, the ribose and RNA and DNA, both of them have approximately the same structure for sugar. This is the group for RNA and this is the group for DNA. As you can see here, DNA contains one less oxygen while RNA contains more oxygen. While the strands, DNA contains double strands and RNA contains a single strand. If we talk about DNA contains double helix structure while RNA contains a single strand, so it does not form helix. But there is an exception, we will discuss it after this table. The next thing is about the location. You know that DNA is present at 3 places in eukaryotic cells. In eukaryotic cells, the DNA is present in nucleus and then in 2 self-replicating organelles. The 2 self-replicating organelles are mitochondria and chloroplast. We have already discussed that mitochondria and chloroplast are independent, they are self-replicating, they have their own DNA, they have their own ribosomes, so they can also live outside the cell. If we talk about RNA, then RNA is present inside the nucleus and also in the cytoplasm. We will discuss about types of RNAs, and there are many types of RNA that are in cytoplasm. The next thing is about the function. You know that DNA actually stores genetic information while RNA helps you to express that genetic information. There are many types of RNAs and all of them will help you in the process of expressing that information that you have stored in the DNA molecules. The next is about stability. Obviously DNA has double strands, that's why DNA will be more stable, while RNA has a single strand, so obviously it will be less stable, right? It is just like a stair, like, you know, if you have a stairs, then the stairs would be more stable. As you can see here, this is a stair like this, it would be more stable. Why is that? Because it contains 2 supporting bamboos while RNA contains a single bamboo, so that's why it is less stable. So that was about DNA and RNA. If we talk about the types, then you need to know that DNA has only one type, that is in front of you, while RNA has many different types. So let's discuss about the types of RNAs. So before moving on to the types of RNA, we discussed about that RNA contains single strand. Yes, but there is an exception. The exception is transfer RNA. Well, actually transfer RNA also contains single strand, but it folds around itself and it forms double strands at some regions, and they are called as loops. There is an alpha loop, there is a beta loop, and there is also a third tertiary loop that contains codons and anticodons sections. So that's why you can say that RNA is not always single stranded. There is an exception, that is transfer RNA. So transfer RNA folds around itself, and what does it forms? It forms loops, and those loops contains some double stranded characteristics. While the other trap that you have to be really careful about is that DNA is only in the nucleus. As you have already discussed about that DNA is also present in mitochondria and chloroplast. If we talk about DNA in prokaryotes, right, in bacteria, the DNA is present in cytoplasm, right? So DNA is present in nucleus in eukaryotes, while DNA is in the cytoplasm in prokaryotes. So you have to be really careful about that too. So you have already discussed about difference between prokaryotes and eukaryotes and their DNA already. So I hope you haven't forgotten that. If you have like still think about like what is the prokaryotic DNA, what is eukaryotic DNA, go back to your lecture, revise them again, so you don't have to be confused about these concepts. So if we talk about types of RNA, there are around like 8 types of RNAs, and they might be really confusing for you. So I will do and make the thing easier for you, and that is you just have to know about these 3 different RNAs. They are the most important ones: messenger RNA, transfer RNA, and ribosomal RNA. So they are the most important ones. You'd only have to memorize them and their functions but you have to know about the other messenger RNAs, about the RNAs, but you don't have to go really deep into them. You don't have to know every single thing about them because they are not relevant to your IMAT exam. I have written them in front of you so that you don't miss any information, but I recommend you to go towards messenger RNA, transfer RNA and ribosomal RNA in great detail. So if we talk about messenger RNA, as you can see, the name is saying that it helps in messaging, right? So what does it do? It actually carry the genetic information. It carries the message from the nucleus to the ribosome, and in the ribosome you will make protein by translation. So to make proteins, the information is in the DNA, right? So what messenger RNA will do, it will take the message from DNA, and from DNA it will take the message to the ribosomes, and ribosome will make the proteins. So messenger RNA is the messenger. If we talk about transfer RNA, the name is saying that it is transferring something. It is like a bus, or you can say a tractor, or that transfers material from one place to another. So what does it transfer? It transfers amino acid. You know that messenger RNA took the information and the blueprint to make proteins, while transfer RNA will gather the materials. What is the material to make proteins? That is amino acid. So transfer RNA will carry amino acids to the ribosomes to make protein. If we talk about ribosomal RNA, so you can say that ribosomal RNA are the machinery to make protein. Like ribosomal RNA are the basic component structures for ribosome. They are the machinery. We have already discussed in your ribosomes chapter that ribosomes are the factory for protein making, while ribosomes RNA are machinery. There is difference between factory and machine, right? So ribosomes are the factory and ribosomal RNA are the machinery to make proteins. While if we talk about smaller nuclear RNA, micro RNA, and then you have other types of RNAs, you can still take a look at them, you can read them, but they are not that highly relevant. You can still give them a read, take a screenshot, and then we will move to next topic. The next topic is about the central dogma. So here is the process and the position and the topic where your molecular biology will actually start. So your molecular biology starts from central dogma. So central dogma was given by Crick. And what is actually central dogma? Central dogma actually means it is the process of how genetic information actually flows and works in your body. So in your body, the genetic information is doing a lot of work, and we will summarize all of those things in central dogma. So what is central dogma? The first thing is about DNA. So what will happen to the DNA? The first process that actually happens is replication. So the first step is replication. After replication, you will have a copy of DNA, and after you have a copy of DNA, the next process will be RNA synthesis, that is transcription. So transcription is the process of conversion of DNA into RNA, and then after RNA synthesis you will have translation. Translation is the process of making of proteins. So this is the process of central dogma. The first process is replication. You make a replica or a copy of DNA. After you have that copy, from that copy you will make a dRNA molecule. After that RNA molecule has been made, it is called as transcription. After transcription, the next process is translation. So what happens in translation? In translation you make protein molecules. So this is the process of central dogma, and we will go deeper into this topic in our incoming chapters. There is a dedicated chapters on replication, translation and transcriptions. You don't have to be worried about these topics. We will go into a deep learning for these topics. So this is the classical central dogma that we just discussed about. Next is topic about chromatin and chromosomes. You have already know that in a non-dividing cell there are structures or smaller DNA structures that are called as chromatin. So chromatin will be in a non-dividing cells, when the cell wants to divide, it will be converted into what? Into chromosomes. As you can see, this is a chromosome molecule. This is the centromere, and we will discuss about chromosomes in detail in your cell cycle chapter. So just take a screenshot, study about the basics. I won't be going into detail because this is a introductory chapter. So you have to know that there are two types of structures that contains DNA. The first is chromatin and the other is chromosome. So chromatin actually is present when the cell is in non-dividing stage, but when the cell is in during mitosis and during meiosis, the chromatin will come together, they will condense together and they will form chromosomes. We will go really deep into chromatin, into chromosomes in cell cycle chapter, but I won't be going into that detail. So just take a screenshot, and now you know what are chromatin and chromosomes. Let's discuss about DNA of prokaryotes and eukaryotes. So first of all, the prokaryote actually contains circular DNA, right? And the DNA is actually present in the cytoplasm. Why is that? Because prokaryotes does not have a proper nucleus. That's why the DNA is present in the cytoplasm, while eukaryotes contains linear DNA, and the linear DNA is present in the chromosome molecules, and it is the chromosome is inside the nucleus. If we talk about histone proteins, then prokaryotics contains no histone proteins, but eukaryote does contain histone proteins. If we talk about ploidy, then prokaryotic DNA are usually haploid, while eukaryotic DNA are usually diploid. If we talk about introns, that we will discuss about in our future lectures, what are introns, what are extrons. Then in prokaryotic DNA they are absolutely absent, but in some rare cases they are do present, but in eukaryotic DNA they are always present because obviously introns are important. If we talk about prokaryotic DNA, it is smaller, while eukaryotic DNA is much larger in humans. If we talk about extra DNA, yes, prokaryotic DNA has extra DNA, and that is called as plasmids. So what are plasmids? Plasmids are the extra DNA that are present in prokaryotic molecules. So plasmid actually contains genetic information for the survival of prokaryotes. For example, plasmid will have antibiotic resistant molecules, right? And antibiotic resistance information will be in plasmid. They also have anti-pesticides and these kind of survival information, so that prokaryotes or bacteria can survive in harsh conditions. While eukaryotic DNA also contains extra DNA, and you already know that they are not present in the nucleus, but they are present in self-replicating organelles like mitochondria and chloroplast. If we talk about replication, so in prokaryotic DNA the replication only have a single origin, that is called oric, while if you talk about eukaryotic DNA, then they have multiple origins of replications. So if you have any questions about intron, about replication, about ploidy, about histones, don't worry about it. We will discuss about replications, about introns, about ploidies, about histones in our future lectures in detail. So for now, just memorize these terms, memorize this table, take a screenshot that is in front of you. Like we have done the basics about introductory chapter, like what are the things that we will study and the basics that you had to know. So this was all about the first introductory lecture, and then we will move on to our next topic, that is going to be our replication, translation and transcription chapters. So that was all. Let's move on to next topic, that is replication.